students, in the previous class I have explained you about the Griffith experiment, and thereafter I also started in the earlier part. DNA is a genetic material. In that I introduced this was experimental proof that is given by first time Hershey and Chase, Alfred Hershey and Martha Chase experimentally proved DNA is only the genetic material. And yesterday also I introduced you about how they started their experiment, and once again I start. In their experiment they have grown the bacteriophage in two different media. They have grown the bacteriophage. Bacteriophage means it is a T4 bacteriophage, means it is a virus. This is a virus, and that structure of the virus also you studied in the first PUC class. That having external coat, this having an external coat, it is a protein. This is the external coat, it is a protein. See, this is a bacteriophage diagram, almost just like a satellite shaped structure. And this is a protein, external coat, that is called a capsomere, and inside there is a DNA. And that is why the bacteriophage is a DNA virus. See, this bacteriophage is grown in two different media. There is one in radioactive phosphor and another one in radioactive sulfur. The bacteriophage which is grown in a radioactive phosphor having radioactive DNA because the phosphor present in the DNA, and the bacteria which is grown in a radioactive sulfur and they having a radioactive protein. Outer protein is made by the radioactive protein, radioactive sulfur. Once again I repeat, the bacteriophage which is grown in a radioactive phosphor having radioactive DNA because the phosphor present in the DNA. Similarly, the bacteriophage which is grown in a radioactive sulfur and they having a radioactive protein because the protein contains sulfur. So two different the virus. Now further it is allowed to infection. See, a radioactive sulfur labeled protein capsule and a radioactive phosphor labeled DNA. Got it? So two different bacteriophages are allowed to two different bacteria, very bacteria, E. coli, and so this is bacteria and this is bacteria. To this bacteria radioactive sulfur labeled protein capsule containing bacteria infect, and to this bacteria radioactive phosphor labeled DNA containing bacteriophage infect. If it is infection ಅಂದ್ರೆ ಅದಕ್ಕೆ ತಾಕ್ಸೋ ಅಟ್ಯಾಚ್ಮೆಂಟ್ ಕೊಡೋದಕ್ಕೆ ಇನ್ಫೆಕ್ಷನ್ ಅಂತ ಕರೀತಾರೆ. And then they were blended in blending ಅಥವಾ blender machine mix ಮಾಡ್ತಾರೆ, blended in the blender machine. And if it is so, here see, if it is so, blending if it is taken place, and in this blending process what that DNA present in the both inside the protein capsule is entering into the inside the cell. This is bacteria cell. Bacteria means cell, remember, inside the cell. So already bacteria also having its own DNA. Bacteria ಮೊದಲು DNA ಕೂಡ ಇದೆ, old DNA ಕೂಡ ಇದೆ. ಈಗ viral DNA is also entering into what? The bacterial cell. Then it is undertaken for the third process, centrifugation. ಈವಾಗ centrifugation ಮಾಡಿದರೆ this bacteria is separately centrifuged and this bacteria is also separately centrifuged. If it is a centrifugal or it is centrifuged and later it is a result is observed, and that result shows that here no radioactive sulfur detected in the cell, but radioactive sulfur detected in the superdiamond of the protein. Got it? Seriya kalpana. Then a radioactive phosphor detected in cells, no radioactive detected in a superdiamond. From this what you understood? From this exactly is what you understood is only the DNA molecule entered into the cell, not the protein. Only the DNA molecule entered into the cell, because here virus, bacteriophage, this is the bacteriophage, and this bacteriophage containing only two components, DNA ಇದೆ ಮತ್ತು ಪ್ರೋಟೀನ್ ಇದೆ. ಇದರಲ್ಲಿ ಯಾವುದು ಒಳಗೆ ಬಂತು? ಬ್ಯಾಕ್ಟೀರಿಯಾ ಸೆಲ್ಲಿನ ಒಳಗಡೆ ಬಂದಿದ್ದು ಯಾವುದು ಅಂದರೆ ವಿಚ್ ಈಸ್ ಅ ಟ್ರಾನ್ಸ್ಫಾರ್ಮ್ಡ್. which is transferred and which is a transformed ಅಂತ ಕೇಳಿದ್ರೆ only the DNA is a transfer, not the protein. Protein ಹೊರಗಡೆ ಇರ್ತದೆ. Protein remain as it is in the same place after the blending. After the blending and the centrifugation, protein gets separated. But what that DNA present inside the protein, that is entered into the what? Here cell, bacterial cell, and later it is come to know that here what the DNA, this DNA is a radioactive DNA. Because phosphor labeled radioactive DNA is radioactive phosphor detected in cells, radioactive phosphor detected in the cells, but no radioactive detected in the supernatant. Supernatant andre protein. Supernatant means what? Protein. Andre what exactly mean? This protein capsule not entered into the cells. Got it? This protein not entered in the cell, but it's only what DNA. ಒಳಗಡೆ DNA ಇತ್ತಲ್ಲ, ಆ DNA ಮಾತ್ರ ಒಳಗಡೆ ಬಂದಿದೆ. Only DNA entered, protein remain outside only. Supernatant ಅಂತಂದ್ರೆ protein remain outside only, but its DNA is transformed. But this is radioactive because it is grown in radioactive phosphor. That is why it is a radioactive phosphor label. And this is grown in radioactive sulfur label. But radioactive sulfur, of course, radioactive sulfur label only in the protein, not in what? The DNA. But even then, that DNA, from here also DNA entered into the cell. But here, both the DNA is original DNA is also there, and this DNA is also entered. This DNA is also entered, of course. But here, this DNA is not radioactive phosphor. Radioactive DNA ಅಲ್ಲ. ಯಾಕಂದ್ರೆ ಆ radioactive DNA ಸಂಬಂಧಪಟ್ಟಿರೋದು ಇಲ್ಲ ಇಲ್ಲಿ. ಯಾಕಂದ್ರೆ ಸಲ್ಫರ್ ಲೇಬಲ್ ಇದೆ. ಸಲ್ಫರ್ ಲೇಬಲ್ ಆಗಿರೋದ್ರಿಂದ ಪ್ರೋಟೀನಿಗೆ ಮಾತ್ರ ಅಟ್ಯಾಚ್ಮೆಂಟ್ ಆಗಿದೆ, ನಾಟ್ ಟು ದ DNA. But as it is, this DNA is also entered. So from this experiment it is understood that in both the case, viral DNA entered into the cell, but one DNA is radioactive, another DNA is not radioactive. So thereby it is once again Martha and Chase come to know that DNA is only the genetic material, not the protein. Why? Because the biochemical characteristic feature was studied by the McCarthy, Oswald Avery and Macklin. They were to have a little bit of confusion. Either protein is a genetic material or the DNA was a genetic material. And for that answer was given by Martha and Chase by conducting this experiment using bacteriophage and bacteria. Bacteriophage means virus. Bacteriophage means virus and the bacteria. So this experiment, I think you understood, better you understood. And if it is asked for the examination for the 5 marks question, you have to write same. So but you need not require to draw here this diagram because I have already given the diagram containing notes. I think once again you refer the same. I think you understood. Very easy. So there is no any difficulty in this. And from this experiment, Martha and Chase finally come to know that DNA is only the genetic material. DNA is the only genetic material, so not the protein. So this is about the experiment. This is about the experiment. Now, next part of this one is, according to your syllabus, properties of genetic material. Properties of genetic material, and that is particularly DNA virus versus RNA. DNA versus RNA. Yes. From Griffith experiment, Martha Chase's experiment, it is come to know that DNA is the main genetic material, and that has a transforming principle, and that the basic knowledge we already got from the 2 experiment, and also the biochemical characteristic feature of this genetic material also studied by the various scientists, and they also come to the conclusion saying that in our body transforming material is there, and that is the DNA. Now here the DNA versus RNA, as we are saying that DNA is a genetic material. Now if it is a question is asked, which is the first, either DNA is first or RNA is first, because our previous knowledge says that no, RNA also act as a genetic material in the absence of DNA. That you should remember. Sometimes RNA also act as a genetic material in the absence of DNA. So that exactly means if it is asked a question, which is a first genetic material, RNA is the first genetic material, DNA is the first genetic material. According to the science, the first genetic material was the RNA. First genetic material was RNA, and that will be it is a single stranded, and you know that. And the beginning of the evolution of the RNA, DNA, beginning of the evolution of the DNA itself start from the RNA. So that is why here two different type of viruses are there. One is RNA virus. RNA virus are primitive, and another one is a DNA virus, and it is evolved. ಅಂದರೆ RNA DNA ಆಗಿ ಕನ್ವರ್ಟ್ ಆಗಿದೆ. During the course of the evolution, RNA only converted to DNA, and if the same this RNA, if another strand comes and it become what? DNA. No, so means during the course of the evolution, RNA is only converted to DNA. So it's RNA virus converted to DNA virus, and further it got what? The nucleus. Further it got nucleus, and thereafter it got what? The cell. So then it becomes the cellular component. So origin of life. How the steps involved in the course of the evolution. So during the course of evolution, biological molecule, origin of life and evolution also you studied, and the next chapter follows same. During the course of the evolution, large number of complex biological molecule like protein, carbohydrate, lipid, plus this DNA and RNA play vital role in the formation of the cell and the cellular component. Similarly, here before the formation of the cell, there was an RNA world was there. RNA take part much in the course of the evolution of the DNA. Basically, RNA is a genetic material, and when the RNA is converted to DNA, in the future evolution ಆಗಬೇಕಾಗಿದ್ರೆ DNA begin to produce RNA. Understood? In the future evolution, DNA is only converted into RNA. That process is called a transcription. You studied. DNA only converted into RNA, and that RNA what catalytically very active and what unstable. ಹಾಗಾಗಿ DNA ಮುಂದೆ ಏನು ಮಾಡ್ತು ಕೇಳಿದ್ರೆ DNA ತಾನೇ RNA ಏನು ಮಾಡ್ಲಿಕ್ಕೆ ಸ್ಟಾರ್ಟ್ ಮಾಡ್ತು, ಕನ್ವರ್ಟ್ ಮಾಡ್ಲಿಕ್ಕೆ ಮಾಡ್ತು, ಮತ್ತು DNA RNA ಕಡೆಯಿಂದ ಕೆಲಸ ಮಾಡಿಸ್ಕೊಳ್ಳಿಕ್ಕೆ ಸ್ಟಾರ್ಟ್ ಮಾಡ್ತು. You got it this point? In what during the course of evolution, RNA converted to DNA, and when the DNA take part in the the central portion of the nucleus, means central portion of the cell, and in the next coming evolution, during the evolution, here DNA begin to start RNA, and from RNA it started to carry its one catalytic reaction, because RNA is highly unstable and also has an ability to carry the reaction, catalytic reaction, the enzymatic reaction. So that is why here the beginning probably, even RNA world also says same, the beginning probably the RNA is the first, and RNA only begin to convert into DNA, and when DNA is converted into RNA, and at that time all the different type of catalytic reaction, the etc. begin, and that play very important role in further evolution of the living organism. Okay, now return back to this properties of genetic material. If it is a genetic material, if any genetic material, what its main characteristic feature? Here it should be able to replicate. If you are considering a material is a genetic, and that genetic material must have an ability to replicate itself. You know that DNA also replicate. DNA replication, you know. DNA also replicate. DNA replication, you know. Correct? DNA also replicate. And when does the DNA replication take place? The DNA replication takes place during the interface of the cell, and that to be in S phase. Correct? That you all know, studied. And during that, it try to what replicates its number of copy, and thereby it can share its genetic material from one cell to the another cell. ಹಾಗಾಗಿ ಡಿ ಎನ್ ಎ ಆಸ್ ಎ ಜೆನೆಟಿಕ್ ಮಟೀರಿಯಲ್ ಆಗಿದ್ದರೂ ಕೂಡ ಅದು ರೆಪ್ಲಿಕೇಶನ್ ಆಗಬಹುದು. ಇಫ್ ಇಟ್ ಇಸ್ ಎ ರೆಪ್ಲಿಕೇಟ್, then only it can share its inherited property to the next generation, and that is very important process. Then chemically it should be very stable. Chemically it should be very stable. Very stable ಅಂದರೆ at any cost ತನ್ನ ಅಸ್ತಿತ್ವವನ್ನು ಚೇಂಜ್ ಮಾಡ್ಕೊಳ್ಬಾರ್ದು. ಭಾಳ ಸ್ಟೇಬಲ್ ಆಗಿರಬೇಕು. ಮನಸ್ಸು ಚೇಂಜ್ ಮಾಡಬಾರ್ದು ಅಂತ ಹೇಳ್ತಾವಲ್ಲ, ಹಾಗೆ ಒಂದೇ ನಿರ್ಧಾರ ಅಂದರೆ ಒಂದೇ ಡಿಸಿಷನ್. ಆ್ಯಂಡ್ ಇಟ್ ಶುಡ್ ಬಿ ವೆರಿ ಸ್ಟೇಬಲ್. ಕೆಮಿಕಲಿ ಇಟ್ ಶುಡ್ ಬಿ ವೆರಿ ಸ್ಟೇಬಲ್, ಆ್ಯಂಡ್ ದರ್ ಶುಡ್ ನಾಟ್ ಬಿ ಎನಿ ವೇರಿಯೇಷನ್, ಆ್ಯಂಡ್ ಇಫ್ ದರ್ ಇಸ್ ಅನ್ಸ್ಟೆಬಿಲಿಟಿ ಇಸ್ ದರ್, ಆ್ಯಂಡ್ ದಟ್ ಕ್ಯಾನ್ ನಾಟ್ ಕ್ಯಾರಿ ಇಟ್ಸ್ ಓನ್ ಪ್ರಾಪರ್ಟಿ ಟು ದ ನೆಕ್ಸ್ಟ್ ಜನರೇಷನ್. So that is why this is also one very important characteristic feature of this. That is chemically it should be very stable, and then it should give more scope. It should give more scope for slow changes or evolution. As we know that when mutation takes place, different type of mutation also you study. Sometimes it is any change in the level of the chromosome, level of the DNA, level of the gene, level of the base pair, point mutation, all that you studied, all that is responsible for here change, and that change may be a slow change or fast change, or whatever it may be, and that is responsible for the evolution, and evolution help in making the new species. That is what we call as origin of new species. This is the third important characteristic feature of the genetic material. So genetic material. Fourth important character of the genetic material is, and it should be able to express itself. It should be able to express itself a Mendelian character. It should be able to express itself a Mendelian character. What is that Mendelian character? See, Mendelian character already you studied, the passing of the trait or character from one generation to another generation, from plant to the plant, or animal to the animal, or cell to the cell, in its particular ratio is 3:1, 1:2:1, like this. So that type of Mendelian character, it has to be here shared in the next generation, and that is also another important property of the genetic material. So these are the four important property of a genetic material. So first one is it should be replicate itself, and that play a very important role in the sharing the genetic material. Chemically it should be very stable and should not change its identity or its chemical component, etc. And that is also very important. It should be scope for evolution. Time to time evolution ಆಗಲೇಬೇಕು. ಯಾಕಂದರೆ environment change, habitat change, so many changes that is taken place in our body also, and time to time it just undergone for evolution, but that doesn't mean that it change its identity change ಮಾಡೋದಿಲ್ಲ. identity change ಮಾಡಿದ್ರೆ ಅದು ಅನ್ಸ್ಟೇಬಲ್ ಆಗಿರ್ತದೆ. ಹಾಗೆ by maintaining the stability, a slight changes that is taken place in DNA sequence, and that is a scope for the evolution, and then itself has to maintain the Mendelian character, and that is also very important characteristic feature of this. So these are the important characteristic feature of the genetic material. Now with this you are going to study next one is the RNA mode. RNA mode. Yes, I need not require to tell about again this. So in the beginning only I told you DNA versus RNA. If it is asked which is the first, RNA is first or DNA first, generally our basic science says that RNA is probably first in the nature, and when during the course of the evolution RNA only is converted to DNA, and RNA is catalytically very reactive